So I think we'll wait until 5 after, and then
Sure.
we'll go ahead.
You know, people are hurrying from other parts of campus, right?
That's true.
I want to say that there's a couple of sort of perks of COVID that I was talking about with my colleagues. One of them, it's really hard to be very late for meetings, right? You're just, you know, you just have to get yourself to sign off and sign on. And the other one is it's easy to learn student names now because their picture is always paired with a name, right? So before, if you ever struggled with student's name, now it's really, really easy. So, you know, gotta look at the positive side of things here.
Right. I think we can, we have about 26 people, so we're a few minutes after 4, so I think we can go ahead and get started. So it is my absolute honor and pleasure to introduce today today's seminar speaker, Dr. Soha Hassoun. And so I had a pleasure of meeting Soha at a conference, Metabolomics Association of North America, and I was very impressed with her research approach that she's going to talk to us about today, and the work she's doing. And so I was very pleased when she accepted our invitation to give a seminar. My deep regret is that it's not in person, but hopefully one day we can correct that when the pandemic is over. And so Dr. Hassoun got her undergraduate degree in electrical engineering from South Dakota State University, and the master's degree from MIT, followed by a PhD from the Department of Computer Science and Engineering at the University of Washington in Seattle. And so she is a professor and in the past the chair of the Department of Computer Science at Tufts University. And so she is using machine learning approaches to study and develop tools for synthetic and systems biology. And so she's also working in the area of metabolomics. And so today she's going to talk to us about various machine learning solutions that she developed for addressing the challenges in the field of metabolomics. Thank you so much for accepting our invitation. And without further ado, please proceed.
Thank you so much. I would like to thank everybody for coming for my talk. I know there's Zoom fatigue, and it's very hard to sort of pay attention for a long time. So I'll try to make the talk as interactive and as entertaining as I can make it. And as Ella said, I'm really excited to be here. I wish it was in person, but it's not. And I hope for better times in the future. So I would like to start out talking about this favorite quote of mine. This quote is from Jensen Huang, who's the co-founder and CEO of NVIDIA. NVIDIA is a company that started out in 1993 making processors for gaming and multimedia applications. So NVIDIA quickly evolved into the business of making GPUs, or graphical processing units, around 2001. In GPUs, the same program can run in parallel on multiple streams of data. And this type of hardware is very appropriate for graphics card, where you're trying to update pixels on the screen simultaneously to maximize the experience for the gamers. What's really exciting about and relevant about the NVIDIA hardware and GPUs in general is that GPUs not only do graphics well, but you could repurpose them to perform complex computations required by AI and machine learning algorithms. So I just want to put this quote in perspective. This was stated in 2017 in reference to the state of software. At the time and currently, very few lines of software in the world implement AI algorithms. All what Huang was saying is that once various disciplines start using more AI algorithms, AI will become the dominant form of software. You might say, what does AI or machine learning have to do with metabolomics? Well, it turns out that the GPU hardware, like the one from NVIDIA, is enabling novel software solutions that will impact on how we do a number of things in life, including metabolomics and biological and clinical data analysis. So this is sort of, let me get everybody on the same page slide. Everybody's familiar with the central dogma of biology. And the stuff that I'm interested in is actually at this level of metabolomic networks. So when I talk about network here, I have metabolites A and B making C. They're catalyzed by a particular enzyme. And together, all these make a network connection. So metabolomics is obviously very important because the DNA gives you the blueprint of what the cell can do, but obviously metabolomics is actually telling you what the cell is actually doing at any particular point in time. And we've already seen some remarkable work on how metabolomics is allowing us to take a biomarker or a panel of biomarkers and make them clinically relevant or applicable for studying cellular processes. In a recent work published in Autism Research, scientists at NeuroPoint DX, in collaboration with others, reported new findings from the Children's Autism Metabolome Project. They identified unique metabolic signatures in over 50% of the children with autism in the study. This is an important step towards the goal of developing a panel of metabolomics tests that could be the basis of a biological screen to analyze the risk of autistic spectrum disorders. There are many other studies, like the ones found in the gut, related to the gut microbiota or to exercise, that have shown metabolomics effective in identifying biomarkers and linking them to health and disease. So really, if we want to capitalize on this technology, the metabolomics technology, we have to be able to interpret metabolomics data well. And that's really what my lab is trying to do, is look at machine learning and try to advance the interpretation of metabolomics data. So for my talk today, I'd like to tackle 3 problems. The first one is, how do we use deep learning networks to do metabolite annotations? And then I would like to talk about how you determine pathway activities and metabolite annotations using something called Bayesian learning. And the last part of my talk is just going to talk about enzyme promiscuity and how it relates to metabolomics and how we could use it, how we could use our knowledge about enzyme promiscuity to advance metabolomics. I'll spend most of my time talking about the first 2, and I'll spend just a little time talking about the third item. So metabolomics is concerned with measuring the masses of small molecules, and I'm sure this crowd is very familiar with the small masses. Of course, here I have a scale just to remind people what we're trying to do with metabolomics. So in untargeted metabolomics, where I'm concerned about biological discovery, I'm measuring everything possible that I could possibly measure in the sample. I measure thousands of molecules, the masses of thousands of molecules. And what I'm trying to do is figure out what I'm measuring. And it turns out to be a super hard problem. So imagine that I measured a molecular weight of 89.0247 grams per mole. There are many molecules that have the same exact weight. So these have the same atomic composition usually, but differ in molecular arrangements. So the biggest challenge in metabolomics is annotation, which involves assigning chemical identities to measured masses. So some very, very, very clever people said, hey, collecting just the mass over Z, is the masses of the measured molecules, is not really sufficient to identify the molecules we're measuring. So often mass spec is applied into 2 phases. In the first phase, we take the sample and we ionize it and separate all the molecules. And you could use a gas or liquid chromatography to do this first step, or you could use MS for the first step. And in the second phase, what we're doing is we're blasting these molecules with ions and some energy and causing the molecules to fragment. And the mass spec is actually measuring, sort of recording what's going on with the fragments. So now, instead of just working with one molecular mass that I measured, I can work now with the spectral signature of the molecule. So now that we have more information to determine the chemical identity of what we measured, the problem turns out to be still difficult. Here's a couple of molecules. They have the same molecular weight. And it turns out that if I blast them through the second phase, and this one actually is not second phase, but it turns out that their spectral signatures are very, very similar. So if you look at these molecules here, the signatures of them just vary by some of these lower intensity signals. So it turns out, as I said, this problem is very difficult and challenging, and the work in this area kind of falls into two categories. Either we could take the measured mass and we could look it up in a spectral database. Okay, and if I have a match, then I know the identity of the molecule. If I don't have a match, then I'm basically at a loss. And of course, due to the experimental effort required to build these databases, the databases are usually small in size and do not cover much of the metabolome. There's other computational techniques for annotation. So there's techniques such as CFMID, CSI, finger ID, and METFRAG. And what they try to do is they try to generate a spectra of some candidate molecules. And then we find the one molecule whose spectral signature most likely matches to the measured spectra. So even though people have been working on the problem for quite a bit of time, the annotation rates remain low, where only a small fraction of measured data is annotated. We have a recent study that shows, with across many studies with spectral lookups, you can only get like 7% of the measurements annotated. So from a machine learning perspective, and I just want to remind everybody that I'm a computer scientist, not a biologist. So I think about problems in a computational sense. So when I see a problem like this, I think about a translation problem. I could think of mapping a molecule to its spectral signature. And we could think of this as a simulation problem, where the mapping is effectively simulating the fragmentation process. We could also think of this problem as mapping the spectral signature to the corresponding molecule that generated the spectrum. And as I showed before, similar molecules have similar signatures. So we do not often measure every molecular fragments. So the problem of going from spectra to molecules is actually a difficult problem, and it's known as an inverse problem in many fields of engineering. I personally very much like thinking about this translation paradigm. I could actually think clearly about these problems from a computational perspective, and I could imagine how existing machine learning techniques and recent advances in machine learning developed for other fields, such as natural language processing and social network analysis, can be used to solve these sorts of problems. And I'd like to point out, even though my talk today is about AI and machine learning to solve these sorts of problems, we're really at the very, very beginning of using AI and machine learning effectively in this area of metabolomics and in terms of clinical and biological data sort of discovery and analysis. So because we're sort of really early here, what we wanted to do is we want to start at a very basic level and describe how to use a neural network to solve this forward simulation problem. So I'm concerned with mapping molecules into spectra. So again, I start out with a simple question. Is it possible to use these neural networks to map molecules into spectra? A neural network is simply a layered network of nodes. In this image, the yellow nodes form a layered network that is 4 layers deep. So 1, 2, 3, 4 layers deep. Each node is performing a calculation based on a parameterized function. The outputs of these nodes are typically numbers between 0 and 1. The inputs to each node are based on the outputs from the prior layers. The input to the network is these nodes on the red. I put the input on this side, and the output is coming out on the right, out of the blue nodes. This type of architecture is often referred to as a feed-forward neural architecture, FFNN. So how do we make this neural network work for our molecule spectral combination? Well, the data set that I have consists of molecules and spectra. So I'm going to use the data that I have to sort of train this model to kind of spit out these spectra when I give it a particular molecule. So the goal of this training that I'm going to do is to learn the parameters that are computed at the output of the blue nodes. And I would like to make the output look as much as possible as my data that I'm using for training. So this type of feed-forward network only understands vectors as input and vectors as outputs. So here at the output vector, I could bin all these spectra into bins, and I could say that for each one of these bins, I'm trying to predict the relative intensity of the peaks in those bins. So that's a vector, and it has different values. And we typically use a bin size of 1,000, and each one of them corresponding to or a small portion of M over Z. So what do we do with the inputs? Well, it turns out that we could represent molecules as fingerprints, as vectors. So the fingerprints are simply binary vectors that indicate the presence or absence of a particular group, functional group, or a ring. And there are many types of fingerprints. So now the neural network could actually be trained to map the molecules, sorry, the fingerprints into the spectral output. So in 2019, Jennifer Wei at Google Brain and other researchers published a paper based on using these feed-forward networks to create additional spectra that they could use to augment their GCMS spectral libraries. So we use this model as a baseline and we improve over it. Okay, and the question is, how can we improve over it? If you look at the step where the molecules are represented, anytime that I go from a molecule to a fingerprint, I basically use some information. So what we try to improve of this baseline is actually how do we improve the representation of the molecules so we could capture more information about them. So we can then take advantage of the structure of the molecule. We can think of the molecule as a graph where the atoms, where an atom is a node in the graph and a bond is an edge in the graph. So this node could correspond to carbon. This edge could correspond to a double bond, for example, to another atom. And it turns out there's a class of techniques called graph neural networks that are designed specifically to take advantage of graph structures. One particular type of GNN is called a graph convolutional networks. And they work by propagating information from the neighbors of a particular node to the nodes themselves. And this information propagation is repeated for each node in the graph and repeated multiple times. If you're familiar with convolutional networks or CNNs, this is very similar, but for graphs. So that's sort of one way that we tackled this problem, is to use this GNN. Another thing that we did is we looked at adding what's called attentional modeling. So it turns out that the contributions of every neighbor of this particular node may not be equal. So what we do is we add something called attention modeling, where we actually learn the relative weights of the neighbors when we're trying to learn the representations of each node in the graph. So now our model for mapping molecules to spectra is updated to replace the feed-forward neural networks with the graph neural networks. So this is an image of a graph neural network. So we no longer need to use the fingerprints. And what we're really doing is we're actually learning and mapping from the molecules to sort of a new embedding, a vector, and then we add a small layer that maps that learned embedding of the molecules into this predicted spectral output. So we've minimized the loss in terms of removing the fingerprint. So let me tell you a little bit how we evaluated the performance of the graph neural networks. So in our test data set, we have spectra and we have known matches. Okay. We know the molecular formula of each real match. So now we could go to PubChem, which is the biggest molecular database, and we could come back with possible candidates that match to the formula of the real candidate. So my GNN will then generate the spectra for each one of these candidates. And then I could compare the spectra that was generated by the GNNs against the spectra of the molecule that I measured in my test data set. And out of all of these, I figure out which of these spectra best matches my measurement in the test set. And then I could go back and say, oh, the molecule that generated the spectrum is the best one. It's my best match. So again, what I'm trying to evaluate how good this GNN is doing, one thing I have to evaluate is how well the spectra match. But ultimately, I want to see how well it does in picking the correct candidate. And hopefully it would pick the best match here. So here's how we kind of did the experiment. What we did is we trained and evaluated and data from the NIST data set. This is for LCMS, so liquid chromatography followed by mass spec. And we selected a subset where we kind of knew something about the precursor types, and we limited sort of adducts selection. So we ended up with 6,289 molecules in our training set, and we actually used about 1,500 molecules for our validation test set. The validation set allows you to select the best model among all the variations of the models that we tried for both GNN and the feed-forward neural networks. Again, because this is more of an art than a science, designing this neural networks, you end up trying lots and lots of things. So the validation set is really saying, pick the best model out of everything. And then what we do is we say, okay, now we could test on an independent set that has nothing to do with the training or the validation set. And for that, we saved 1,000 molecules. And for these 1,000 molecules, we went to PubChem and retrieved the candidate sets associated with these 1,000 molecules. What I show here is a histogram showing the distribution of the number of candidates. Many of our test molecules have hundreds of candidates, and it turns out that the average number of candidates was roughly 1530. So using this candidate set, we can now evaluate how well GNN and FFNN perform. Before I get into the details of that, I want to show you a sample spectral prediction. It's this green one right here. So here we have something called a mirror plot. This is what I measured, and this is what our tool is predicting. And you could see that this is a close match. So I have to admit, when I saw this one, I had to ask how my grad student who's working on this project, if this was the best plot that he had generated. And he did say that it's one of the best, but I made him go back and look at many, many more. As a matter of fact, we looked at the ones that did really well and looked at the ones that did really badly. And there was a lot of them that had high similarity between the actual measured spectra and between the predicted spectra. Okay. So the accuracy of the spectral prediction is not the only metric that predicts the performance of the annotation tool. It turns out that the number of molecules in the candidate set plays a big role. So here I compare the performance of the GNN and feed-forward networks, assuming a different sizes of the candidate sets. I basically take the distribution that I showed you, the histogram distribution of the candidates, and I downsample it until I get a target average number of candidates per molecule of 50, of 250, and 1,000. Okay. So then I score the similarity of the generated spectra against the NIST spectra, and I rank the candidates based on the similarity score. So in each of the figures, I'm actually plotting the top K rank. This top K rank simply means that the likelihood of hitting the target molecule when I look among the top K candidates. For example, when I have 50 candidates and I'm looking at and I'm using the GNN, which is this dark line right here. And when I'm looking at the top 10 ranked candidates, I have an 80% chance of hitting my target molecule. Okay, so out of the 10 that I test, there's an 80% chance that I'm going to actually find it. So here's the number for candidates are 50. Here they're downsampled to 250 and then to 1000. So there's some trends that I want you to observe. First, in each one of these cases, the GNN, the purple line, outperforms the green line, the feed-forward neural networks. And of course, they both outperform just a random guess. And I want to point out here is the performance of both the GNN and FFNN is really very much dependent on the size of the candidate set. As the candidate set size increases, you're going to see a decrease in performance. I also want to point out that it's not just the size of their candidate set that influences this top K rank performance. We designed another experiment to sort of evaluate the quality of the molecules in the candidate set. For each of the 1000 test molecules, we selected various size candidates of 50, 250, and 1000. And we handpicked the candidate sets as follows. So for example, in the case of the size of 250, using the fingerprint similarities on the candidate set, we determined the most similar 250 molecules and the least 250 similar molecules to the test molecule. And we use those 250 molecules as a test set. So in this sense, we actually made up a candidate set that has the least similar molecules and a candidate set that has the most similar molecules. And we repeated this for the candidate size of 50 and the candidate size of 1000. And for each one of these plots on the top and the bottom, the darker line shows the performance when the candidate set is not so similar to each other. And the lighter color shows what the performance is when the molecules are super similar to each other. So the top three plots show how the performance of the feed-forward neural network on the least and most similar sets. And the bottom one shows the same experiment with using the GNNs. So the takeaways here, in both cases, the performance is a function of the similarity of the candidate set to the target test molecules. It seems that the feed-forward networks are less sensitive to the similarity than GNNs. In each of the feed-forward networks, the performance gap between the most and least similar sets are narrower than they are for the GNNs. But keep in mind, the GNN outperforms the feed-forward networks, so there's a sensitivity and performance trade-offs at play. So here's a summary of sort of the first part of my talk. We talked about how to use GNN to map molecules into their spectra. And then GNN outperforms the feed-forward networks due to the learned molecular representations that we're doing with the GNNs. So the performance results are a function of the number of candidate molecules as well as their similarity to the target molecule. And this is really pointing out that we as a community need to be more consistent in the way we evaluate metabolite annotation techniques. We could not really compare our work with any of the other tools because they train on different data, they evaluate on different data, and we can't really recreate exactly the same computational setup to compare apples with apples. And that's a really big deal when you're working in algorithm and machine learning. You have to do comparisons fairly. I would like to here thank my students, Hao and Shimon, and Professor Liping Liu, who's my collaborator. And there's a short paper that kind of describes this work on the archive. Okay. So in this part of my talk, I would like to talk about the interpretation of untargeted metabolomics data in the biological context of the sample. And you know what? I'm going to take a 1-minute break and say, does anybody have any questions about the prior part? About the GNNs or the spectra or the results?
Hi, this is Chelsea. I was wondering, how do you estimate how many, the size of the data you need to train a valid and trustworthy model?
It's an excellent question. You know, there's this big notion when they started working on machine learning that you really needed huge data sets. And it turns out that you do really need to have huge data sets, but sometimes you only have what you have, right? It's not like you could invent new data sets, even though there's techniques called data augmentation that help you kind of increase the size of your data set. But it turns out there's like something called overfitting. So you train your model, and you just don't want it to memorize all the answers for you. So when you train, even if your data set your training data set is small, you just want to make sure that your model is not overfitting. Okay, so you could train with a smaller set. You're not going to get the same quality performance that you would get with a bigger set, but you just have to be careful that your model did not get very specific to that particular smaller data set. Does that answer your question? Thank you. Yeah. So this data issue is always a problem, right? We always want to have bigger data. This is the sad part with the state of biological data. There's so many inconsistencies. There's so many contradictions. You end up, by the time you clean up your data, you toss out half your data set because of various issues. So it's really, really problematic. But we do the best that we can without spending all our time on curating data. Other questions? Great question, Chelsea. All right. So in this part of the talk, whoops, I'd like to talk about the interpretation of untargeted metabolomics in the context of the biological sample. So in this case, what I'm going to do is I'm going to have some measurements, untargeted metabolomics. And what I would like to know is I would like to answer two questions. Which metabolites are present in the sample? That's always the question that everybody wants to answer. And the second one, I want to know which pathways are active or which pathways produced the observed measurements. And in this case, I have a very precise definition of a pathway being active. I will say a pathway is active if the likelihood that that pathway generated the observed measurements is above a particular user-defined threshold. So our goal in this work is to answer these two questions using something called probabilistic modeling. Sorry, I missed a slide here. Okay, so there's several techniques that people have used to determine pathway activities. There's something called overrepresentation analysis. I'm sure some of you working in gene expression data or metabolomics have seen this before. And here we want to know if a pathway is enriched in measured metabolites to a degree different than expected by chance when compared to other cellular pathways or those in a reference sample. And enrichment analysis also encompasses technique that compute observed metabolite centrality and connectivity. And these metrics here kind of reflect how important the metabolite is in a pathway or a network. The earlier works that interpreting metabolomics in a differential fashion. So for example, I compute the enrichment ratio of a perturbed sample as compared to a reference sample. These techniques sometimes assume that the metabolites have known identities. And one technique called mummy-chug actually bypasses this metabolite annotation and computes the functional enrichment of modules relevant to others in the sample without actually annotating the metabolites. And that's sort of the direction that we would like to take. I like drawings. They always make sense to me. So in our work, we don't really want to focus on differential analysis between a perturbed and a reference sample. What I want to do, I want to use all the data that I have, okay, as well as the network structure that I could get for the sample to interpret metabolomics data. So to better understand the data, basically, we're going to capitalize on our understanding of the uncertainty in mapping measurements to metabolites to pathways. So here I have a snippet of a pathway, of two pathways, pathway 1 and pathway 2. Each pathway is associated with several metabolites. So pathway 1 is associated with these metabolites, pathway 2 is associated with these metabolites, and so on. And you could see some metabolites like J2 is actually associated with pathway 1 and pathway 2. When performing metabolomics, we observe several masses, or M over Z's. Okay. In this picture, the masses are represented as squares. Some measurements, such as W1, can only be attributed to one metabolite, J1. Because J1 is the only one associated with the pathway 2, we could potentially say that pathway 2 is active due to the presence of this measurement and its related metabolite. Some masses, like the one here with W2, can correspond to multiple metabolites like J2, J3, which can be associated with multiple pathways. We can all agree here that we have more certainty in attributing W1 to the activity of pathway 2 than the certainty we would have in attributing W2 to the activities of either pathway 1 or 2 or both. So the idea of this work is simple. I would like to use probability to model the uncertainties in mapping the measurements to the metabolites and to the pathways. To solve this problem, we use probabilistic modeling. This involves several steps. First, we define what's called a probabilistic model using random variables. Our model captures relationships between pathways, metabolites, and their masses. Then we make assumptions about the distributions of the random variables. And then once we have the model and the model parameters, we can then form hypotheses and calculate the probabilities of the hypothesis given the observed data. And this is a pretty standard machine learning fare. We can use Bayes' rule to compute such probabilities. So I'm going to give you sort of the visual of what I just said, because I think sometimes it is dense to sort of take that in all at once. So again, what we do is we introduce 4 random variables in our case here. So we have a random variable that tells me whether a pathway is active or not. A value of 1 says the pathway is active, and a value of 0 indicates that it's not active. You can imagine if you have a cancer pathway or some sort of biodegradation pathway, and you don't have a reason for those pathways to be active, they will have 0 activity. While other things like a TCA cycle, it's very likely that it will be active. So in this case, we have as many random variables as we have number of pathways. The second random variable indicates if a metabolite was generated due to a particular pathway. The third set of random variables indicates if a metabolite was present in the sample. There are as many random variables here as there are metabolites in the network. Finally, there's a set of random variables that indicates if a particular mass within the bin centered on each mass within the network is observed. There are as many such mass bins and corresponding number of variables as there are masses in the model. So I want to point out here, this random variable, the observation, is the only random variable that's actually observed using the mass spec, while all the others are considered latent or hidden variables. I don't know anything about them. I don't know if they're there in the sample or not, and I need to learn more about them through this model. So to continue with this, to define our probabilistic model, we assign distributions for the various random variables. So we assume pathway activities denoted by letter A has a Bernoulli distribution over a parameter lambda. That is, we assign each pathway a discrete probability distribution of a random variable, which takes the value 1 with probability lambda and the value 0 with probability 1 minus lambda. Metabolites within the active pathways are equally likely to be generated by the pathway with probability mu. And random variables associated with each metabolites in the samples denoted by M are derived based on the contribution of individual Z variables associated with each pathway metabolite parent. Finally, the random variable associated with each mass bin in the model is derived based on the observed accuracy, gamma, associated with the mass spec. And if any metabolites associated with a corresponding mass was generated by an active pathway. So in this probabilistic framework, our model parameters are actually lambda and mu and gamma right here. So we can assign the parameters some values. We have investigated the sensitivity of our model to these values. The model is actually not sensitive to these values because it's the structure of the model that is the dominant factor that derives our inference results. So we use inference. And the way this works is the model we developed represents the joint probability distribution over all our random variables. Using a probabilistic model, we can infer pathway activities and metabolite presence from observed mass measurements. So we calculate two probabilities. First, for each pathway, P, we calculate the posterior probability of the pathway being active given the observations of the masses W. And then for each metabolite in the model, MF, we calculate the posterior probability of the metabolite being present in the sample. So these metabolite annotation, the probabilities can be used for annotation. And if the calculated probabilities are above a user-defined thresholds, the probabilities are used to suggest and rank the best metabolite matches for each mass measurement. So I would like to emphasize here that the way that we've used inference in this work is different from other prior works. For example, PubMed assigns empirical formulas given potential formulas using probabilistic modeling. So this is different than prior work. So we could carry out the inference using something called Gibbs sampling, which utilizes sampling to approximate our probability distribution. In this work, we use something called collapsed Gibbs sampling. As we marginalized out hidden variables Z and M, the sampler therefore samples only random variables associated with each pathway activity. We achieved significant speedups when we did this marginalization because we had fewer variables, and we could run the inference on the examples that I'll show you in 3 or 4 minutes when we draw 1,000 different samples. So let me jump to sort of some results here. We built this tool called PUMA, probabilistic modeling for untargeted metabolomics. And we analyzed 3 data sets, a synthetic benchmark data set and 2 data sets that we collected from our collaborators. I want to talk a little bit about synthetic data sets. And this is really, really important when we're doing things in biology. Often, we actually do not have the ground truth. So when we say one algorithm works better than another algorithm, you actually have no ground truth that tells you, indeed, that is the case. So even though we have metabolomics data sets from many databases such as Metabolites, Metabolomics Workbench, there are currently no untargeted metabolomics sets where they identified the pathways and we can use as ground truth when predicting pathway activities. So what we did is we built synthetic data sets to mimic the biological process, okay, where genes within pathways work in concert and result in enzymatic activities that produce metabolites. So we used something called the Chinese hamster ovary cell to kind of generate these models, and we were able to generate different data sets assuming different pathway activities and different metabolites being active within each pathway. But now, because I have these synthetic data and I actually know what the ground truth, I can actually compare different techniques on this synthetic data set. And I can tell you, I know this algorithm is better than this one. So what we did is we compared the area under the curve for synthetic data sets, and we compared PUMA with this enrichment analysis. I will kind of speed up a little bit to give times for answering questions at the end. Across all our experiments, it turns out that PUMA outperforms the enrichment ratio by roughly 8%. So this tells us that PUMA is able to recover pathway activities better when using enrichment ratios. We also used it for metabolite annotations, and we outperformed a tool called Bayesan that aggregates various data sources like mNetLin and HMDB and other annotation tools, and also network connectivity. So here's a quick summary of part 2. We developed this probabilistic model for metabolite annotation and for analyzing pathway activity. And we used inference to compute the various activities. And we show significant improvements over other tools for both computing the enrichment ratios and also for identifying metabolite identities. And we have a paper that's published, so you're welcome to look through that. I want to move on to the last part of my talk, and that's the short part, is we wanted to look at enzyme promiscuity and metabolomics. So in this case, there's two areas relevant to metabolomics that actually motivated us to look at enzyme promiscuity. You could generate possible metabolites that don't exist, say, in databases by applying promiscuity prediction on known molecules within the sample. Okay? And as I showed earlier in the talk when we did the metabolite annotation, the quality and the size of the candidate set matters. So what we're doing in our work here is we're using enzyme promiscuity prediction to help us generate a relevant biological candidate set. The other work that we're doing in this space is we wanted to analyze unexpected enzymatic products when you do engineered cellular host. So typically when you design a synthesis pathway that you add to a microbial host like E. coli, you see lots and lots of unexpected enzymatic products. And if we knew something about promiscuity, when we measure those products in the cell, we're able to sort of explain where this data came from. So let me tell you about our enzyme promiscuity techniques. Our group has been working on this very actively. And a few years ago, we came up with a tool that's based on rules. So I learned enzyme X performs bioreaction B. So I could look at the pattern biotransformation and I could learn what the pattern is and I could apply it to new molecules and I could predict new products. And actually all the work that I showed before here was really based on proximal. And as we got really, really good results on these studies, we're like, wait, proximal is not really that awesome. We need to come up with better ways of predicting promiscuity. So we have several machine learning techniques that are in the works, and some of them are close to being published. For example, we use recommender systems like the ones that you use for Amazon or Netflix, where you recommend products to users. We actually do recommend molecules to enzymes and vice versa. We also use natural language processing on protein sequences to predict interactions with molecules. We also have a way of using it, and I'll explain these two other problems that we're working on in the next couple of minutes. So this is one of the first ideas that we started working on and got very excited about with machine learning. And I just love this idea. And it's very, very simple. I'm sure you're all going to be able to relate to it. There's a powerful concept in machine learning called graph embedding. The idea is super simple. Nodes within the graphs are more connected should appear closer together in a multi-dimensional space. So here I have a cartoon. I have nodes 1, 2, 3. They should appear closer together in this two-dimensional space with x1 and x2. So again, 1, 2, and 3 are closer together here than say 1 and 9, right? Which are really far apart in this graph. So we could build a reaction network from databases such as KEGG or Brenda. And then what we could do is we could ask questions like this. What's the likelihood of node 1 being connected with node 4? Meaning, if I have compound A and compound B here, what's the likelihood that they're connected by an enzymatic reaction? And I could say, how does that probability compare compared with finding an enzymatic reaction that ties 1 and 9? Well, we could use the distances in the multidimensional space, and we actually train a shallow network to learn sort of what the likelihood of these connections based on these multidimensional representations of the nodes within the graph. So we have some great results, and this work was just published in Bioinformatics. The other idea that I want to talk about is using multi-label hierarchical classification to predict if a particular enzyme acts on a molecule. So the idea here is very simple. I build a classifier, and the classifier's output correspond to a particular enzyme class. Okay, the enzyme classes are given by the enzyme commission number. So for example, the enzyme commission of this enzyme is given by these 4 numbers. The more, further down along towards the 4th digit, it tells you more specific information about the substrates that the enzyme acts on. So what we did with this multi-label classification is we actually utilized the hierarchy that's inherent in these enzyme commission numbers. And we were able to generate some very interesting results that showed lots of things, such as when you train these classifiers with inhibitor molecules, you actually do better. We also showed that if you do some realistic splits on the data, your performance varies tremendously compared to just doing random splits. So if you're trying to use these kinds of machine learning tools to predict on unseen data, you really need to think about how you're training your model. Okay, so a quick summary. There's great opportunities to utilize machine learning and AI to advance metabolomics and biological and clinical discoveries in general, and the time to do that is now. Our lab is doing some very exciting things, in my opinion, in my students' opinion, to do metabolite annotation, pathway analysis, and enzyme promiscuity prediction to be used in metabolomics, annotation, and other applications. I would like to acknowledge my funding sources, the National Science Foundation and an R1 from NIGMS. And my lab members, this is a picture from the summer during COVID time, are just absolutely phenomenal and incredibly productive and exciting and engaging. I am very lucky to sit between collaborators on the machine learning side. Lipping and Mike are my machine learning collaborators, and the other collaborators are all metabolic or biological engineers or synthetic biologists. I'm always looking for bright and motivated PhD students. So if you're interested or know somebody who might be interested, please let me know. And we're planning a special issue on metabolites with Simon Rogers and Justin Vanderhooft. If you're interested in applying for that, let me know. All right, and with that, I conclude, and I ask you if you have any questions.
Well, thank you so much. I don't know, I would like to, in view of the applause from the audience, thank you so much for the interesting questions. So maybe I can start. So the part that is sort of, the most near and dear to my heart is the metabolite annotation. And so do I understand correctly that in your sort of approach and your simulated data set that you created there, so you're assuming the identities of the metabolites are known, and the pathways consist of certain number of metabolites, and obviously not all metabolites are measured, and sort of, vice versa. So not everything can be mapped to pathways. Is that correct?
Yeah.
Did I understand that correctly?
Yeah. So for that, you're talking about the synthetic data set.
Right.
For the second part of it.
Is that how you built it?
Yeah.
Yes, I'm talking about this.
Yeah, we basically assume is the pathway active or not active. And if the pathway is active, sure, we say which, some portion of the metabolites within the pathway are observed, right? Or
right, exactly,
active, are actually active, like present, right? Because not every metabolite in every pathway is actually active, right? It depends on what you're feeding the cells, what's going on, right? Biologically speaking.
Absolutely, yeah.
And then we also assume that we are not measuring everything that is there as well.
Right.
And
so then my question is,
yeah, go ahead.
No, go ahead.
What's the question then?
Okay, so what I'm wondering is, and you're comparing the performance to traditional enrichment analysis type methods, right? And so what I do know is that compared to, for example, enrichment methods that are being used for, say, gene expression data in metabolomics, there are various metabolites such as NADP, ATP, and so on and so forth, like basically, that map to multiple pathways, right?
Yeah.
And then there are also, metabolites that are very highly correlated by virtue of being the substrate and the product of adjacent reactions or something like this, which usually has some effect on traditional enrichment analysis methods. How would you say your approach kind of handles that sort of situation? Does it give an advantage or?
So these are all very, 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 very good questions. Obviously, you're really into the thick of it. So what we did with the popular metabolites, right? These are hub metabolites. They're commodity. They're in every part of the cells. So what we did is we actually removed them. So we're not trying to measure any NADH, right? I mean, it's not very informative, right? So we actually removed we have a list of cofactors that we have gotten from our biological engineering operators, and we kind of ignored that. So the other question is that some metabolites are occurring right within multiple pathways.
Sure.
Right? It's normal, right? So the point of the study here is that what we're doing is we're trying to find the probability of the metabolite being generated by that pathway. So again, let's say the metabolite is, part of two pathways. If I end up, if both pathways in real life are generating this, I should get equal probability, right? That both pathways were generating that metabolite. If one pathway is not active or just not generating that metabolite, I would hope that the other evidence that I measured, right, is telling me there's chances are higher that one pathway generated it compared to the other pathway. So I really think about what we're doing here, which is different what other people are doing with just the differential analysis. I'm hoping that some of the context of all the stuff that you're measuring is helping me make a better informed decision about what's going on in the cell. And back to your other question is, sometimes you have substrate product relationships, right? We actually do not account, this model that we have for PUMA right now does not account for those relationships, but we have ongoing work to sort of incorporate that in our new iteration of this.
Great, thank you.
Other questions?
Well, if not, I guess, thank you very much again for the wonderful talk. And I understand that you are sending this link and talking to students next. Is that right?
Yes, I believe so.
Yeah, Chelsea and Honey, right?
Yes, I believe so.
Right.
Okay, thank you so much, Paula. It was nice to see you.
So it was great to see you. I will have to follow up with you by email. I mean, I have more questions, but we can do it offline and kind of, I'll follow up. Thank you.
Okay, thank you.
Wonderful.